The next very important topic is oxidative phosphorylation. Oxidative phosphorylation is a biological process in which the ATP molecules, or I can say the energy, is produced in the presence of oxygen. Oxidative phosphorylation occurs in the mitochondria, and oxidative phosphorylation process is divided into two main processes. The first one is the electron transport chain. This is also called as ETC, and the another biological process is citric acid cycle. Citric acid cycle is also called as tricarboxylic acid cycle, that is TCA cycle. ETC, electron transport chains, which is occurs in the inner membrane of the mitochondria, while as the citric acid cycles occurs in the matrix of the mitochondria. This is our structure of the mitochondria. This is the matrix of the mitochondria, and this is the folded inner membrane of the mitochondria, and this one is the outer membrane of the mitochondria, and this is called the inner membrane space, that is space between the outer membrane and inner membrane. In this inner membrane of the mitochondria, there are many carrier proteins, or I can say there are many channels protein. These channel proteins helps in the transport of H positive ions, oxygen, water across the membrane. This membrane also having many proton carriers, that is NAD, FAD, FMN, ubiquinone. All these are the protein carrier. Ubiquinone is also called as coenzyme Q. And this inner membrane also having many electron carriers like as cytochromes. The cytochromes are of many types, that is cytochrome C, cytochrome C1, cytochrome A, cytochrome A3, like that. And in the cytochromes we know that very well, that is there is a Fe metal in the center, which may be in +2 oxidation state, or it may be in +3 oxidation state. So this is the structure of the inner membrane of the mitochondria. These are the channel proteins, and these are ubiquinone, and this is the cytochromes. This ubiquinone and cytochromes are act as the electron carrier, while these channel proteins helps as act as the protein carrier. So first of all, the NADH change into NAD positive, and during this it releases the 4H positive ion in the intermembrane region, that is 4H positive released from the inner membrane to inner membrane space, and during this it releases the 2 electrons. These 2 electrons are transferred to the Q, that is ubiquinone, and FADH2 again change into FADH. During this again the 4 electron is released in the inner membrane space of the mitochondria, and 2 electron release is transferred to the ubiquinone. This electron then transferred to the cytochrome C, and this is fourth carrier or channel protein, again release 2H positive during this FADH conversion into FAD positive. So total 8 or 10, sorry, total 10H positive is released during the inner membrane space. This 10H positive then goes to the ATP synthetase enzyme, which is also present in the inner membrane of this mitochondria. So this 10H positive form the 1 ATP molecules with the help of enzyme ATP synthetase. And these 2 electrons, the electrons removed from here in the presence of 2H positive, which is present in the matrix of the mitochondria, to form water in the presence of 1 atom of the oxygen, or I can say half molecule of the oxygen. We can understand this complex process in a simple way. Supposed to be this is the food molecules or our substrate. This substrate first goes to convert into A, and it releases 2H positive. These 2H positives are accepted by NAD positive, and it form NADH plus H positive. During this, 1 ATP molecule is produced. Then this NADH again convert into NAD by releasing its 2H to the FAD positive. It form here FADH2. These two hydrogen goes to this FAD, it will form FADH2. And during this whole process, the electrons are removed. Two electrons are removed during this process. Then this FADH2 again change into FAD. That is, this is oxidized form, this is reduced form, this is reduced form, this is oxidized form. When hydrogen is attached, then it is reduced form. When hydrogen is not attached, it is the oxidized form. Then FADH reacts with ubiquinone UQ or coenzyme Q to form UQH2. This two hydrogen is transferred to the UQH2 and UQH2 is reduced in UQH2. UQ is reduced into UQH2. Then UQH2 transfer these two hydrogen to the inner membrane space and again change into UQ. And during this electrons are released. These electrons are accepted by the cytochrome B. First accepted by cytochrome B. And we know that very well, the cytochrome having the Fe plus 2 and Fe plus 3 in its center. So it's changed into the reduced form of cytochrome B, and this reduced form is Fe in plus 2 oxidation state. Cytochrome B reduced form again oxidize by transfer these two electron to the cytochrome C1 and it forms cytochrome C1. This is our Fe plus 3. This was the oxidized form of cytochrome C1, and after accepting the two electron from cytochrome B, it changed into the reduced form of cytochrome C1. Then cytochrome C1 again transfers two electron to the cytochrome C, and cytochrome C then further reduce in plus 3 form to plus 2 form. And during this step, when cytochrome B releases its electron to cytochrome C1, another ATP molecule is also produced here. Now the cytochrome C again oxidized by transfer its two electron to cytochrome A3. So here this is the oxidized form of cytochrome A3. It accept the electron from cytochrome C and change into the reduced form. Then cytochrome A3 donates this two electron to 2H positive and form water in the presence of 1/2 molecule of the oxygen. So in the electron transport, the sequence of the cytochrome is cytochrome B to cytochrome C1, then cytochrome